Am I audible?
Yes, ma'am.
Yes, ma'am.
Yes. Okay. Good evening, all. I am Dr. Padma Priya, Professor and HOD, Government Medical College, Bongol. I'm very thankful to the IAMM-TAPC members for giving this opportunity to present this topic, Vaccines 2.0, the Reverse Vaccinology Era. By the end of this session, the participants should be able to explain why reverse vaccinology was developed and compare the traditional and reverse vaccinology. Listen. Describe the workflow of reverse vaccinology, and they should be able to differentiate reverse vaccinology 1.0, pangenomic reverse vaccinology, and reverse vaccinology 2.0. And at the last, they should be able to recognize the landmark success and future directions of the reverse vaccinology. And the infection control, it is achieved through multidimensional approach, but disease prevention is the most efficient approach of the health. The development of vaccine is the milestone in the prevention of the disease. First, we should know what is vaccines and what is vaccinology. What is the vaccines? A biological preparation. It is designed to stimulate active immunity, active acquired immune response, and confers the protection against the infectious diseases. And here it is the tool for active immunization, and it should induce the humoral and cell mediated immunity, and it establishes the immunological memory. So what are these vaccines? What does it contain? The vaccine contains a substance that is antigen, active substance that is called antigen, and it is a part of the microorganism, or it is one of the structure of the microorganism, maybe a toxin or maybe a protein or maybe a polysaccharide, like that. So the vaccines, what is vaccinology? What is vaccinology? The multidisciplinary science engineering of vaccine research, development, manufacturing, evaluation, and public health implementation is the study of these multidisciplinary sciences, the vaccinology. So, the vaccines, it contains an agent. This agent stimulates the immune system to recognize this agent as a threat and destroys it and recognizes it further whenever it comes into across, and destroys these microorganisms associated with the agent that may, if it encounters in future also. So this is the importance of the substance or an agent in the vaccine. And next is the we have achieved the major achievement of immunization is smallpox eradication and polio eradication, and coverage of 86% of the vaccine coverage globally. And what is the public health impact of vaccination? So the vaccination prevents the diseases, it reduces the mortality and morbidity, and it is providing long-term immunity, and it is used as a preventive and therapeutic it is used as preventive and therapeutic function, and there is no drug resistance for these vaccines. And this is the time scale of vaccines. So in 1796, the smallpox vaccine, it was invented by the scientist Edward Jenner, and averting the infection by um, by collecting the material from the cow. And later it was the toxoid vaccines were introduced, and followed by polysaccharide vaccines. And there was a first licensed hepatitis A vaccine was developed in 1992, and human papilloma virus vaccine in 2006. And finally, the COVID-19 vaccine was approved in 2020, which is a Pfizer biotech, so which was a breakthrough. And we have saved from this COVID-19 pandemic because of this COVID-19 vaccine, which was developed in 2020. And he is the scientist, he is the Craig inventor, and he has first completed he has deduced the free living organisms bacterial genome, that is Haemophilus influenzae. So why if he identifies this bacterial genome sequence for free living organism, what is its impact? Its impact is a shift from the empirical vaccines to rational designing of the vaccines, and it also access to the entire antigenic repertoire. What is this repertoire means? Antigenic repertoire means the total T cell epitopes and B cell receptors of an individual's immune system is the entire antigen repertoire, and it is the foundation stone for reverse vaccinology 1.0. And this is the historical milestones. The historical milestone is the classical vaccinology 1796. What is the technology they used? Growing of the microorganisms and live attenuated and killed vaccines were developed. And here the antigen is identified via culture only. And reverse vaccinology, the concept was identified in 2000 by Reno Rapoli, and it is mainly genome sequence based technology, and it is the focus is mainly on vaccine candidate antigen prediction, and finally it is using the computer tools and computer tools, and finally it is producing the vaccine. That means without the growth of the microorganisms in the lab, the first it is the genomic approach we are using in reverse vaccinology, and developing the vaccines after predicting by computer tools the predictive candidate vaccine antigens. And next is the reverse vaccinology 2. In 2002, it was the concept has come. Here the focus is mainly human monoclonal antibodies were isolated and they were cloned. And next it is combined with the structural based antigen design. So here the integration of human immunology and structure based antigen design for development of the designing of the vaccines. Whereas in the reverse vaccinology, the focus is mainly on the genome sequencing, predicting the potential vaccine candidate antigen and developing the vaccine only. Here in the reverse vaccinology 2, the isolation of the human monoclonal antibodies, and it is integrated with the structure based design, antigen design, and finally it has developed the it has instructed to design the vaccine antigens. And next is the classical vaccinology. And the classical vaccinology followed the rules of Pasteur, Louis Pasteur, which was the isolate, inactivate, and inject. And this isolate, inactivate, and inject. Here the vaccine development completely relies on the culturing of the pathogen in the lab. And the antigen is discovered empirically, and it is only a trial and error. And here the vaccine is attenuated, the developed vaccine is attenuated, inactivated, and toxins were developed. We will discuss in the next slide. And here this is the culture of the organism. Isolate the pathogen and cultivate in the lab, and the antigen is separated. That means purification of the antigen. That means antigen, specific antigen is separated by mechanical and physical routes. And what is the inactivation, and what is that attenuation? Inactivation means it is killed. The microorganism is killed. It will not replicate. So where it is used in immunocompromised people who require repeated boosters of vaccines, boosters of doses. So it is the inactivation, and only the humoral immunities participates in the inactivation. Whereas in the attenuation, the whole microorganism is weakened and is through serial passages, and this weakened pathogen will replicate, whereas inactivated pathogen, it will not replicate. Here it replicates and it produces the immunogenicity. So after this, it has completely, the pathogen is isolated, antigen is separated, it is inactivated or attenuated, and finally the vaccine should be prepared by adding the formulations. What are the components of the vaccine? It is the preservatives to prevent the contamination after the vial is opened, and add the stabilizers to maintain the antigen's integrity. The stabilizers may be sugars, amino acids, or proteins, and the surfactants are added. They prevent the aggregation and they improve the suspension's stability. Vaccine component suspension's stability is improved by adding the surfactants. And next, diluent like distilled water is added to ensure the correct concentration. And finally, the most important one is adjuvant. The adjuvant, it enhances the immune response. It prolongs the antigen exposure. So the adjuvant is often added to the separated antigen to produce the efficient and safe vaccine. And after the production of these vaccines, we have to do the immunogenicity testings. That means whether it is the separated antigen with adjuvant is able to produce the protective immunity or not. We have to test in the animal models like mice, rabbits, or human primates, and it identifies the antibody titers, neutralization, protection, and also at the same time we have to evaluate the safety also, which is very important. After doing these trials directly, we cannot release. We have to do the preclinical trials and the clinical trials, and then finally phase 4 and release the lot. So what is this preclinical trials? We are using the animal studies only for immunogenicity testing, toxic evaluation, whether it is toxic or not. It is identified in the preclinical stage, and then the investigational new drug, it undergoes phase 1, phase 2, phase 3 human clinical trials. So this phase 1, we use 20 to 100 volunteers, and we will identify the safety of the vaccine and the dose of the vaccine, optimizing the dose of the vaccine and immunogenicity of the vaccine. Whereas in the phase 2 trials, we are identifying the 200 to 1000 volunteers, and we are extending this extended safety, immunogenicity, and dose optimization. We are identifying the phase 2 trials. In the phase 3, it is from the general population, not from the volunteers. It is from the general population. So we are using 1000 to 10,000 plus population. Here we can see the extended safety monitoring, and efficacy and dose optimization, whether it is proper or not. And then we go for the biological license submission. So biological license submission, we have to submit to the US Food and Drugs. They will come and they will inspect our clinical trials, and they give the license. And after this, we have to go for phase 4. Phase 4 is mass vaccination. It is post-licensure monitoring. So manufacturing monitoring, and what is that? The safety monitoring just before the release of lot. Post-licensure, we have to monitor all these things, and marketing surveillance also be done, and then release the lot. This is the classical vaccinology. What are the strengths? It is the smallpox eradication and eradication of the poliomyelitis. And in many countries, there was a mortality due to infectious diseases like measles, diphtheria, tetanus in olden days. But with the invent of the vaccines for against these infectious diseases, it has come to negligible. The mortality has come to negligible. So the established proof for the vaccines that they are working is known, and it is the laid foundation for the modern precise vaccine development. And then what are the limitations? It is culture dependent approach. So we have to think of whether any high risk microorganisms are there, biohazardous microorganisms are there, which are difficult to handle. So we cannot culture those organisms. And there are microorganisms which cannot be cultivated in the lab. For those microorganisms, the vaccines cannot be prepared. This is one limitation. And empirical antigen selection and limited to immune dominant antigens and long and unpredictable development timelines. That means the preclinical phase only it will take more than 5 to 10 years. And clinical phase, each phase 1, 2, 3, 4, it is taking 3 years time. So the finally it is taking more than 25 years of time for the lot to release. So it is a long timeline. And reversion to virulence, sometimes accidentally in the lab while doing these trials or cultivating, it may reverse, the reverse virulence may be reversed, and high risk of biohazard handling. And what are the, it has reached to a dead end. So it is unsafe for capsule based strategy, and subcapsular protein approaches were very insufficient, and decades of research without a viable vaccine, because many of the vaccines are failed that by traditional method between the preclinical trials and lot release or clinical trials. So it is very what is that so much of work is done but without a viable vaccine. So it is a new strategy is urgently required at, at this age at this stage. So what are the pathogens that challenge the classical vaccinology? Antigenically variable vaccines like Neisseria meningitis and molecular mimicry with the human tissues or host tissues, and fastidious or unculturable pathogens like intracellular bacteria. They will evade the immunological responses and immunological memories. They will evade, they will escape. So for those things it is a challenge, and intracellular persistence and immune evasion is once again a challenge. Then the critical question comes into picture. If the organism cannot guide the antigen selection, can the genome provide the answer? Can vaccine discovery begin in silico? So a new direction in vaccinology was about to emerge. And there is a paradigm shift from the organism based discovery of traditional methods to genome driven design of reverse vaccinology. So he is the scientist Reno Rapoli. He introduced the concept of reverse vaccinology, and the first vaccine which used the reverse, which was developed by using reverse vaccinology is Men B vaccine, that is meningococcus serotype B vaccine. So it is a rapid and competitive strategy in the development of vaccines, which utilizes the complete pathogen genome sequence for antigen screening. What is this? What is this? I means first, in the we have to do the genome sequencing of the pathogen to screen the antigen selection, then identifies the potential vaccine candidate antigens by computer tools, and it detects the surface microbial proteins, and based on this genome level analysis the vaccines have been developed. So what is the main approach here? Genome approach is the first approach, which is and also along with computational tools we are predicting the vaccine candidate antigens, and then developing the vaccines is the basis of reverse vaccinology. Genome analysis, genome sequencing, and then computational tools, and identifying or predicting the potential vaccine antigen is the basis of reverse vaccinology. So it is a new method or modern approach which uses the computational technologies to identify the vaccine targets from the pathogen genome. We are already sequenced the pathogen's genome. From that sequenced genome, we are predicting the potential vaccine targets by using the computational tools. So that has transformed the vaccine designs. So It has enabled the discovery of potent vaccines against a wide spectrum of infectious diseases. So during the traditional vaccine development, over a century, we have only 25 to 30 vaccines only. But with the advent of the reverse vaccinology, we can develop the potent vaccines against wide spectrum of infectious diseases. Unlimited number of infectious diseases, we can find unlimited number of vaccines by using this technology. So it is mainly, we have to remember, it is the genomic approach. Antigen is predicted by computational tools and a vaccine is prepared. Okay, next, why it is called reverse vaccinology? In traditional approach, the pathogen is cultivated, isolate the specific antigen, and then vaccine is developed by using this antigen. But in the reverse vaccinology, we are demonstrating the genomic sequencing and analyzing the genome by computer tools and predicting the antigen and then vaccine. So we are not we are not going into the development of the pathogens in the lab. We are skipping that aspect and completely genomic approach we are doing first. So this is the reverse approach. That's why it is called reverse vaccinology. And what are the core principles of reverse vaccinology? In the reverse vaccinology, there are the 4 terms we should know. So what are those 4 terms? It is the genomics. What is the genomics? What is defined genomics? We should know these basic terms. Without that, we cannot understand the reverse vaccinology, computer tools, all these things. So what is a genome? What is genomic? Genomic is the study of organisms, entire set of DNA, that is genome, including the structure, function, evolution, and mapping of these genes beyond a single gene studies. So it is the heredity, we are studying the single gene studies. But in the genomics, we are going beyond the single gene studies. It is a comprehensive study of organism's entire genome, entire set of DNA. We are studying, including the structure, function, evolution, and mapping. So here the next is the what is this ORF prediction? What are these ORFs? ORFs are open read frames. They are nothing but potential protein coding regions, and they are used as primary targets for gene prediction algorithms. They are nothing but they are all computer analyzing programs. So these gene prediction, we have a prediction algorithm ki this is the target. This ORF is the target. What is ORF? It is nothing but potential protein coding region. And also in silico antigen mining. That means in silico antigen analysis and prediction. Why it is called in silico? In the computer, the major chips and are prepared of the silicon material. That's why they are called in silico analysis. And notice the bioinformatics. The bioinformatics are in study of interdisciplinary field which merges the biology, computer science, mathematics, and statistics to analyze and interpret the complex biological data like DNA and protein sequences. That means we are analyzing the DNA and RNA sequences by using integrating computer science, statistics, and mathematics. We are utilizing all these things, integrating and analyzing the biological data, that is protein, DNA and protein sequences. It is essential for genome sequencing. And next is the proteomics. The proteomics is the mapping of the proteins. The expressed proteins they are mapping. It is done by mass spectrometry, and it is for mainly analyze the protein structure, function, and expression. And finally, we are getting the immunogenicity, that is protective antibodies are produced. This is the core principle. How reverse vaccinology works. This is the overview or It is the basic view, genome analysis, in silico screening, proteomic filter, immune validation, and vaccine candidate. We are discussing each in other slides. So, what is this genome sequencing? We have to do in the reverse vaccinology, whether it is a 1.0 or 2.0, we have to do the genomic sequencing. It is of three types. One is the first Sanger sequencing, second one is the next generation sequencing, third is the long read sequencing. So the first one is only chain termination method. It is also called dideoxy sequencing. So here the chain termination occurs with the dideoxynucleotide triphosphate, which stops the DNA at a specific sites so that the fragments and stretches of DNA are formed. The second one is most important. We discuss in detail the next generation sequencing. It is also called massively parallel sequencing, high throughput sequencing. Here there are 4 steps. First, we have to collect the samples, biological sample like tissue, blood or saliva, and isolate the DNA and RNA and assess the purity and concentration by using the spectrometry and or fluorometry. So we have collected the samples, isolated the nucleic acid, that is DNA, and then purified that nucleic acid. And then second one and most important one is library preparation. See this picture. The first in the NGS, we have to break down the DNA into small manageable fragments. And these manageable fragmentation along with adapter ligation. Listen carefully. This step is very important. Fragmentation and library preparation. So what is this library preparation? Here, the adapters are used for this ligation. What are the adapters? Adapters are nothing but short synthetic DNA sequences that are attached to these fragmented small DNAs. So by attaching these adapters to small fragments, it is enabling this to bind to the flow cells. So we will discuss clearly these adapters by attaching to the fragmented DNAs. The binding will enable to bind these small fragments to flow cells. So what are these adapters are not there? What will happen? If adapters are not there, DNA fragments cannot bind to the flow cells. So no PCR amplification, no sequencing of primers occurs. So what are these flow cells? Flow cells are nothing but glass slide or microfluid chips where DNA and RNA fragments bind, amplify and sequenced. So this is very important. Fragmentation and library preparation. Library preparation means the DNA adapter ligation to synthetic DNA sequences attached to small fragmented DNAs which are attached to the flow cells. In the flow cells only, the library is loaded onto the flow cells. Then in each molecule is amplified to millions and millions in a small cluster area. That's why it is called cluster generation. Whenever the library is loaded onto flow cells, cluster generation occurs. And then these another important point in next generation sequencing is sequencing by synthesis. What is the term sequencing by synthesis? So bases are nucleotides are added to the library strands, and these library strands are attached to the nucleotides are attached to these library strands and detected by the real time. So what will happen? What is the importance? This produces millions to billions of short read sequences. This is important. In NGS, only short read sequences we can identify, we can get. This is the raw material. So next step is the data analysis. In the data analysis, the raw signals are converted into nucleotides. They are called FASTQ files. And mapping occurs. They compare the short rates, which are raw material, and they are compared to the reference genomes and analyze. These 4 steps are NGS. 4 steps are required in the process of NGS. Why we are stressing on NGS means reverse vaccinology 1 and 2, and it is based on the sequencing by NGS. So after the sequencing by NGS, the genome sequence is studied using the bioinformatic techniques. That's why we have we have discussed the bioinformatics. What is bioinformatics in the previous slides? So we will find the genes which are involved in the virulence, pathogenesis, or key biological processes. Why we are analyzing this genome sequence? Only to find these genes which are in virulence or in pathogenesis or key biological process. So what will you predict by analyzing this? We will predict where the proteins are present in the bacterial cell, that is localization of the proteins, and functional domains and motifs within the protein. The motifs and what is the motifs? It is in the bioinformatics only. They are the short sequence patterns in DNA or RNA which holds the biological significance and determine the molecular function. So finally, we are detecting the molecule's function, protein's function, identifying the functional domains and motifs, and finally assess the immunogenicity, whether they are protecting immune responses are present or not. These are all things are analyzed by the bioinformatics, that is computational tools. And next is the proteomics analysis. What are these proteomics? Proteomics are nothing but expressed genes. They are the proteins which are expressed on the surface. So these proteins, they are analyzed by the proteomics techniques, and by using the tools like mass spectrometry, electrophoresis, and microarrays. And immunological validation is the last step in reverse vaccinology 1.0. Here we are testing, after analyzing all these things, we are testing whether this predicted vaccine candidate antigen is able to stimulate the immune response in animals or humans. So this involves the animal models, in vivo or may in vitro also. We can test the ability. So in the animal models, we test the candidate proteins. We are injected into the animal models and assess the immune response and protection. By ELISA, we can detect the antibodies or cytokines. By flow cytometry, we can analyze the immune cells to determine their response to target proteins. So reverse vaccinology in action. So entire thing what we have discussed is in the lab. So how it is helpful to the patients, to the public, how it is useful, that is the action. So here one of the most important first vaccine which is licensed by reverse vaccinology is Bexsero. It is against the Axangium nisera meningitis. The target antigens are surface proteins which are expressed on the surface of the pathogen. This may be OMV or outer membrane, or FH may be whatever may be the targets. They are the surface expressed proteins on the pathogen's surface. And Shingrix, it is the viral vaccine against the varicella zoster. The target is the glycoprotein E. And the parasitic is the Muscarix, which is the against the Plasmodium falciparum. Here the target is circumsporozoite protein. And virus in fungals, mucormycosis, it is in the pipeline. The preclinical trials are occurring now. So only the predicted adhesins or surface proteins are the targets. And reverse vaccinology advantages: rational and efficient identification of the targets, accelerates the vaccine development process. That means in the traditional, it is taking more than 25 years. Here it takes weeks to months only to develop the vaccines. It is a great achievement, and also identifies the conserved antigens. Conserved antigens and across the strains. What are these conserved antigens? The conserved antigens means they are the essential antigens which must be present across the different strains, different serotypes, so that broad spectrum vaccines are prepared. Again, is using these conserved antigens. And it detects the less immunogenic and improves the efficacy. And development of personalized vaccines. What is this personalized vaccine? Some people, they have high risk of contracting a specific infectious disease, or the person may be immune compromised. So the person's genomic sequence is identified, and his immune responses are detected, and they prepare the vaccines for that category of the people. It is, they are called personalized vaccines. Limitations: So it requires large scale genomic data sets and high cost of bioinformatics, and there is a translation gap between prediction and clinical applications. So we have discussed reverse vaccinology 1. It is the genomic approach. You have to remember the final take home messages in reverse vaccinology is the focus is on genome sequencing. Genome approach is the first base, and predicting of the potential vaccine antigen by computer tools to develop the vaccine. So the next is how it is evaluating the reverse vaccinology. So reverse vaccinology evolution is reverse vaccinology 2.0. It is the evolution of genome-based antigen discovery into structure-guided vaccine design strategy. So from identifying the antigens and developing the vaccines to we are designing the antigens, we are designing the vaccine strategies. So we have developed from the traditional way of developing the microorganisms in the lab to we are designing the vaccine strategies. We are designing the antigen structures. So that is the rational, it is the revolution in the vaccine development progress. So what will happen in reverse vaccinology? It integrates the human immunology and the structural biology together, and also it involves the high resolution antigen antibody interaction mapping. So human immunology means we are isolating the monoclonal antibodies, and the structural biology means we are forming the 3D structure of the antigens, 3D structure of the proteins. And what is this meaning of high resolution means even a small base pair genetic variation is there also, it is detected with high accuracy and sensitivity is high resolution antigen antibody interaction mapping. So we will discuss later. What is the structural vaccinology pillars? There are 5 pillars which are epitope specific immunity. The name is indicating immunity again is epitope specific, and neutralizing sites, that means the binding sites. We will discuss in detail. Structural insights, that is atomic level guides antigen design. Prefusion form, it drives the immunogenicity. Engineered antigens elicits antibodies. So what is this structural vaccinology 2? So this structural vaccinology 2 means it is also first identifying the antigens or proteins by genome analysis. The genome analysis is nothing but next generation sequencing, analyzing the genomic sequence of the pathogen, and with the computational tools, that is bioinformatics, we will predict the antigen. So that is the genomic analysis. So the proteins may be surface proteins or secreted proteins, whatever it may be. What is the target selection? The target selection means here the first approach is the isolation of the human monoclonals. That means here what is the important step? What is the core principle? Here the individuals who have naturally developed neutralizing antibodies against an infection or against a pathogen have selected. That means they are naturally developed antibodies by exposure to the pathogen, or the neutralizing antibodies are developed. By studying these monoclonal antibodies, how they are binding to the pathogen, the scientists have identified the exact vulnerable spots that need to be targeted. That means it is very precise. Unlike the prediction of the vaccine antigen in the reverse vaccinology, and here the target is very precise. We are selecting the epitopes, or we are selecting the binding spots that needs to be targeted. So here you can see the human antibodies are present. The antibodies have variable regions. The variable regions are the combining sites to the epitopes of the antigen. So the place where the paratope is binding to the epitope of antigen is the binding sites or vulnerable spots or epitopes that needs to be targeted. That is called target selection. The targets may be bacterial adhesins in bacteria, and what is that, proteins in the viruses. Next step is the structural characterization and epitope mapping. So it is also containing the 4 steps like reverse vaccinology 1.0. Here the step 2 is structural characterization and epitope mapping. Here what is the important cornerstone in structural vaccinology is atomic structure determination. This is very important, and this is the modern rational approach that utilizes the 3D atomic level information to create, to engineer, or to optimize the antigen that triggers the immune response. That's why the atomic structure determination is very important. So these identifying the precise atomic structure of pathogens surface proteins, which are identified in the prior step, genome analysis and bioinformatics use and identify the surface proteins. So those surface proteins atomic structure is identified. So the precise atomic structure of that pathogen surface proteins are identified. Especially they are the epitopes targeted by neutralizing antibodies. That also we have discussed in the previous slide. The neutralizing antibodies, they're going and binding to the epitope of specific antigens. Okay. And the next is the we are identifying the atomic structure determination by cryo-electron microscope, X-ray crystallography. So the combination cryo-electron microscope, it is identifying the overall structure. X-ray crystallography, it is determining the atomic structure of specific binding site, that is epitope. So hybrid approach is very effective. And next is the antigen antibody complex. We know that monoclonal antibodies are going and binding to the epitope of the antigen. So that antigen antibody complex, it is the 3D structure of this epitope site is determined. It is all by the computer tools, bioinformatics. So efficient, stable, and safe structures. These are 3D structures. If you develop a 3D structure, it is efficient, stable, and safe. And next is the epitope identification. So how the epitope is identified? The previously determined is only the 3D structure of the binding site antigen antibody complex. But here it is the epitope identification. That means mapping. That means neutralizing epitopes. They are identified. The exact binding sites, they are identified. So the next is the identification of malleable antigens. The malleable antigens means it is the pre-fusion and post-fusion. Two conditions are there. The virus fusion protein before entering into the any cell is the pre-fusion state. After entering into the cell is the post-fusion state. But after entering into cell, the post-fusion state, it identifies only ineffective antigens or ineffective epitopes. But pre-fusion antigens, they will identify the exactly stable proper antigens or proper sites it will identify. It is called pre-fusion state. So compare it to post-fusion, pre-fusion is very important. So we have to maintain the antigens in pre-fusion stage to improve the immunogenicity. So the next is so we have to lock the antigen in the pre-fusion state to improve the immunogenicity. How we had to lock these antigens in pre-fusion state? So one is by proline substitutions. Proline substitutions. These are nothing but proteins. We are substituting in the antigens. And cavity filling mutations. We are introducing the cavity filling mutations into these antigens to lock the antigen in the pre-fusion state. So another one is addressing the antigen drift. So what is this means? Suppose influenza is their organism. It contains a variable region and a conserved region, the anti. So what is this variable region? The mutations occur in the variable region. The conserved region, it is devoid of mutations. It is an essential aspect. So remove the variable head and you target the conserved stem for proper immunogenicity response. And next is the epitope focused design. So that means some non-neutralizing antibodies will be present. Unwanted antibodies will be present. So according to that only the epitopes are identified. So these unwanted non-neutralizing antibodies, they are deleted. So they are deleted and they are removed. So that the conserved antigens will be enriched. The conserved are important for vaccine development. So the next is the epitope scaffolding. So what is this epitope scaffolding means? Some critical regions are present in the proteins. And these critical regions, when they are grafted onto stable scaffolding of the protein, it maintains the stability of the antigen and it also increases the immunogenicity. So that's why we have to do this epitope scaffolding. And the next is the chimera construction. Chimera construction means multivalent epitopes will be present. They are merged into mosaic or chimeric antigen, which increases the stability and also improves the immunogenicity. So the rational antigen engineering, that means the antigen we have predicted in the first step after sequencing of the genome, we are modifying, intentionally we are engineering this antigen to improve the stability, to improve the immunogenicity, to get the efficient vaccine. Next is the fourth step is the platform integration. So without the platform, we cannot use this modified, rationalized, engineered, stable antigens. So we have to select one platform for its proper functioning. So these platforms are recombinant protein vaccines, virus These advantages are rational, precise basic design, broad strain coverage, improved vaccine stability, enhanced safety profile, and reduced risk of autoimmunity.
Ma'am, sorry to interrupt you, ma'am. The slides are not visible. Suddenly they just went off.
What, Vijayendra? Lights?
The slides are not visible, ma'am. Can you please
Slides
share
are
again?
not. I can
Yeah, just now, like just since 2 minutes.
So now it is.
No, it's not there. Just can you please share again?
Share again. Okay. Sir, 1 minute, ma'am.
Yeah.
ఇది బ్యాక్ చేయాలండి ఫుల్ స్క్రీన్
So, Sravanti, Sravanti, the slides are not visible. What to do now? Minimizing of PPT slide. So, how to do?
They were there till now. Just the last 2-3 minutes, they just went off.
So I have to minimize. Yes, I'll minimize this slide.
You
Is
could
it
just
going-
reshare again. You could just reshare again.
Reshare.
Stop sharing, share again.
Stop sharing. Okay. Share screen again. I have to share. Yes, sir. So, share.
Yeah, now you can know.
Okay. Yes, yes.
Now audio?
Is it visible?
Yeah, it's back. It's back. It's
Okay,
coming.
okay,
Right.
okay.
Thank you.
Yes. Yeah, I'm doing full screen.
Probably just maybe if possible couple of slides back.
So see this advantages. Preclinical validation. Is it
Yeah,
okay,
I think
Vijayendra?
yes. Please, ma'am. Yes.
I have to tell again. In silico evaluation and
Ma'am, we are at the platform.
yeah, we are at the platform, Amma. 1 minute. Yes, from here you are unable to see the slides, sir.
Yes.
Okay, so the platform integration is very important because the modified engineered antigens, intentional engineered antigens for maintain the stability efficacy, we are integrating into standard platforms. They are recombinant protein vaccines, virus-like particles, mRNA vaccines, viral vector vaccines, nanoparticle display systems. Here, preserve correctly folded antigens similar to native structure to improve the immunogenicity, which is very important to remember. And next is the preclinical validation and in silico evaluation, in vitro studies, in vivo animal studies, and domain identification. That means we are using the computational tools and identifying the stability prediction and binding affinity to binding affinity of the binding affinity is also it is studied in silico evaluation. Next, we are doing in vitro studies to antigen testing, stability assessment, and in vivo studies in the animals for safety, immunogenicity, and antibody response. And later, domain identification and safety optimization. We have to remove the immune suppressive domains because they will cause the autoimmune responses, which is very dangerous. So we have to remove the immune suppressive domains so that we will reduce the autoimmune responses. This sequential evaluation ensures the safety and absence of autoimmunity. And reverse vaccinology advantages, 2.0 advantages are, it is very precise basic design. Compared to the reverse vaccinology 1, it is very precise because epitopes are identified and they are used as targets, and we are analyzing and preparing the vaccine designs. So rational precision based design and it targets protective epitopes, and it is a broad strain coverage and improved structural stability by doing the engineering and rational intentional modifications of the antigens. We are maintaining the structural stability and enhanced the safety profile and reduced the risk of autoimmunity. So what are the future directives? Advanced epitope prediction algorithms may come, and protein structure prediction models may come with AI, and immunogenicity ranking systems and accelerated antigen prioritization may come in future. So what are these next generation vaccine designs? So AI is involved in optimization. AI optimization means AI is guiding the antigen optimization. Now, till today, we are predicting the antigens. We are only the identifying with computer tools the epitopes, which is very precise. But later, with our artificial intelligence, we may guide the antigen optimization, and we may get to the universal epitopes, epitope targeting vaccines, pan family vaccines, and we can prepare for any pandemic, because by reverse vaccinology it will may take only a few weeks to months time to prepare vaccines, unlike traditional vaccines which will take years together. So, and next we may use the mRNA platforms. For integration of mRNA platforms, we have identified SARS-CoV vaccine. So, in the future, the mRNA platforms may usage may be more, and more vaccines we may prepare. And what it changed, it has shifted the vaccinology from empirical to rational, and expanded the antigen discovery beyond the culture dependent limits, and accelerated early phase vaccine candidate identification. And why it matters? It integrates the genomics, immunology, and antimicrobial resistance. Everything is integrated, and it expands the scope of modern clinical microbiology. It bridges the laboratory science and public health. So these are my references. What is the take home message? So there is, with the invent of Craig Venter, it has revolutionized the vaccines development progress. And again, with the concept of Renopoli, we have revolutionized. It is a big milestone, the preparation of vaccines by reverse vaccinology, and we can develop any type of vaccines against a widespread infections. So in the future, we can expect the more number of vaccines against all infectious diseases. Thank you for listening, and thank you for giving this opportunity to IAMM-TAPC members, all the members. I am thankful to you. So any doubts or any questions, you are pleased to ask.
Thank you so much, ma'am. That was a wonderful lecture.
And thank you.
Apologies, ma'am. I mean, like we couldn't be there at the beginning. I just came
It's
home
okay.
and then probably the session
It's
started off.
okay, Vijay. It's okay. It is for
Yeah.
PGs, not for the
Yeah.
faculty.
No, I'm sure all the PGs loved it. It's a futuristic topic. That's what people like nowadays. And I must say, ma'am, it was a very nicely made talk, and even the slides were very interesting.
Thank you.
All the PGs, I mean, like I can ask you to put in any questions on the chat box. Madam will be able to take that. We'll take in for 5 minutes, ma'am.
Yes.
One question has come, ma'am.
Yes.
Anything about rabies antibodies investigation? Blah, 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 blah. I don't know. Somebody has asked about rabies antibodies in this.
These antibodies, they're a DNA recombinant.
Examples of vaccines by reverse vaccinology.
Examples of vaccines I have shared. One is the Boxero, which is against the Neisseria meningitis, and which targets the surface active proteins. And another one is one virus, that is herpes zoster vaccine Shingrix, and it targets the what is that? It is targeting the what is that? Glycosides, I think. And the third one is the what is that? Fungus is in the pipeline for mucormycosis, and a parasite, that is the malaria muscarix, which is the target is the sporozoite protein. And another important one is our 2.0, the vaccine is SARS-CoV vaccine. They are the target are the spike proteins.
Almost everyone is thanking you for making it easy to understand, ma'am. People
Okay.
have written it, like complicated topic, but yes, madam made it easy for them.
Thank you. Thank you. Can I stop sharing?
Yeah.
Okay, thank you very much for all the PGs for patiently listening. And I will thank the IAMM-TAPC, all the members for giving this good opportunity to share my knowledge. Thank you very much.
Thank you, ma'am. Thank you so much. Thank you, all the attendees. Thank you so much. Good night.
Okay, ma, thank you. I'm stop sharing. Stop sharing.